sessions. Welcome, Alex. The floor is yours.
Great. Thank you so much for that kind introduction. So today I'm going to tell you a little bit of, I'm Alex from ORIC Pharmaceuticals. So today I'm going to tell you about a case study of our work to ensure diversity in chemical screens by evaluating molecular features. So on the next slide, I'll first tell you a little bit about ORIC Pharmaceuticals, which is a clinical stage precision oncology company with a pipeline targeting multiple resistance mechanisms. We go after traditional cancer drivers, such as our CNS active EGFR HER2 exon 20 inhibitor. We're also exploring emerging solid tumor targets, such as our potentially best-in-class PRC2 inhibitor in prostate cancer. We're also kind of looking at immuno-oncology targets, such as the CD73 inhibitor in multiple myeloma, where we saw single-agent activity, nice safety profile and immune activation, and have a healthy discovery research program where we're going after a PLK4 inhibitor, as well as multiple undisclosed programs. So on the next slide, I'll tell you a little bit about bioinformatics. So we're really contributing insights throughout the biology, exploration, and translation cycle of the drug discovery pipeline. Here at the bottom, we start with understanding the biological mechanism of action. We go to find what is the right gene that we can target and how it's involved in those mechanisms that drive cancer. We find the PD and predictive biomarkers to ensure that clinically we're engaging the target and we can find the right population where our drugs work. We test that in clinical trials, evaluate the data that comes out to find the drug resistance mechanisms, and start the process over again. In general, we're evaluating genomics, primarily multi-omics data, to ensure that we're efficiently transitioning between these stages and can inform every program as we move. But today, on the next slide, I'm really going to focus on a slightly different element and just this one case study where we're looking at early stage programs and how we can create a diverse and efficient chemical screen by looking at chemical properties. So in this case, we're employing machine learning strategies to identify a set of very diverse chemical structures. We have created a method of clustering more than 500,000 molecules to optimize selection of compounds to represent diverse chemical classes. And we're utilizing AI-based techniques to inform structure-activity relationships and suggest chemical modifications that can improve biochemical potency. So on the next slide, I'll start off by telling you a little bit about what we're going after and main idea here. So we have a target of interest identified. So we realize that we need to do a novel chemical screen to actually engage that target. So we really want to assess a novel screening technology. And to do that efficiently, we're only going to sample a subset of the entire chemical library. So here on the bottom left, our partners, they have a diverse compound library. We combine that with a target engagement readout and go through a high throughput screen to find for hit finding and going through the hit optimization process. Now, in this problem, the compound library has been pre-allocated on plates that are fixed. So we can't change what compounds are on what plate. And we really want to maximize likelihood of success. And to do that, we're going to find the most diverse set of plates for screening. Now, we utilize an unbiased machine learning approach to get a per plate diversity score to be able to find that subset of plates that can help us efficiently move the program forward, efficiently screen and move the program forward. And we do this in a library of, with 500,000, more than 500,000 compounds. So on the next slide, I can tell you a little bit about how we go through this process of taking a molecule. In this case, it might be a familiar molecule to some of you. We look at what substructures are present on that molecule and transform it to a fingerprint. So the fingerprint here really captures bitwise information where a cell is gray if a substructure of that molecule is found, is present on that molecule, and it's light gray if it's not present. So that's a simple representation of a molecule through a bit vector. On the next click, we can now take these bit vectors and compare similarity between two molecules. So here we have molecule A, molecule B. They look similar. They are similar. And based on the fingerprints, we can really see how much overlap in terms of their chemical substructures are there, and use that to calculate a molecular similarity score. It's a very simple score. In this case, it's the intersection over the union. It's the Jacquard similarity. In cheminformatics, it's called Tanimoto score, but we'll just call it the overlap or molecular similarity score in this case. And on the next click, we'll just use this molecular similarity score and compute it between each pair of compounds that we have and calculate a per plate diversity score. So next slide. Now, we start off by looking at how similar are the compounds per plate, and we're going to use a simple eigenvalue decomposition to inform that. And here, I think we'll just use a simple example to allow for, to show the intuition of how this works. So on the next click, if we have 6 molecules, compounds A, B, C, D, E, F, and we have a case where all the molecules are identical. When we do our eigenvalue decomposition, we see that all the weight is on the first eigenvalue. Now, on the opposite end of the spectrum, on the next click, have a case where all molecules are distinct and the eigenvalues are, the weights are distributed across all. On the next click, maybe 2 clicks forward, one more. And here we can really see that we can kind of create toy examples and really provide this understanding that we can utilize the size and distribution of eigenvalues to inform the diversity of each plate. The top left, all very similar, have one type of distribution. And on the other side, everything is distinct and has a different distribution. So moving to the next slide, we take and apply this approach to all the plates in our suite. And we do it for 2 types of fingerprints here on the left. We have the Feature Morgan, the RD-Kit layer fingerprints, and generally we can see that they agree. There's a pretty good correlation and agree on the kind of most and least diverse plates. Now, the red plate, which is our least diverse plate, you can clearly see that there are substructures of molecules. So every column and row is now a molecule. The value inside that is the amount of overlap between those molecules. And you can see there are sets of molecules that are very similar to each other. More than 50% of the molecules similar, and we expect it to have a similar biochemical potency, or at least inform on the SAR. The other side, we have a very diverse few substructures, and this really suggests that this method, this eigenscore, it allows us to rank plates and evaluate the within plate diversity. Next slide. We simply calculate the eigenscore for every plate, lots of different fingerprints, and we can aggregate it together. We can cluster it. We can see that they're diverse plates and have a single eigenscore. However, we realize that even though we might have very low within plate diversity, you might still have similar compounds between different plates. So on the next slide, we go about figuring out an approach to find similar compounds that are present between plates. So on the next click, the way we do this is for every compound, we basically ask, what is the most similar compound on another plate? And what is that overlap score? If it passes the threshold, we'll say that those 2 compounds are equivalent. So in the case on the top left, we have our test compound. Let's see for the top one, plate 2. I'm not a chemist, but just by eye, see that there's a lot of similarity, and the score says the same thing. Plate 114, on the other hand, we can visually see that they're different, and that's what we get. Now, we can simply just calculate what is the proportion of equivalent compounds from one plate to another. And on the next slide, that's what we do. We do that across all the plates for all the compounds. And we can see patterns in the data. We see that first grouping of plates are all very similar, then they're not similar, and then some similarity, and it varies after that. We can go ahead and do that on the next click for all the plates across the entire library, do it across all the samples, and start to see patterns that You can see plates that are clearly distinct from everything else, as well as clusters of plates that seem to be similar. Now, with that in hand, we basically have a within plate similarity score, between plate similarity score. And on the next slide, we can just utilize what we've already calculated. So we have two within plate scores. We have this eigenscore within plates. We have the proportion of similar kind of non-self molecules. They're very well correlated. They practically give the same information. You can find them. And we can also use that on the right-hand side where we take the within plate score, look at the between plates, and that allows us to utilize and select what is the most diverse set of plates that are available. We're now able to efficiently sample as much of that chemical space as possible and allow for multiple hits starting points to be able to optimize off of from this chemical screen. Now, on the next slide, there are a lot of possibilities that we're exploring, utilizing, and going forward with. One of them is we have these similarities already. We can now create a network of compounds, try to understand how they can inform, how they correlate, neighborhoods of compounds correlate with different drug properties, how they correlate with biochemical potency. ORIC expertise is really in structure-based drug design, and we're trying to explore how we can optimally use and incorporate AI/ML into that. We found predicting protein conformation, binding site elucidation methods really useful. We're also looking at modern Gen AI type techniques to help design novel drug-like compounds that will push our programs forward and go in and explore novel chemical spaces. So we're kind of continuously testing these different methods internally to move, to incorporate within our chemistry workflow and moving forward. So with that, I'm going to finish off by acknowledging on the next slide. A lot of people from chemistry team, I'm not a chemist, I don't know that much about chemistry, but we had a lot of help here on that aspect. People from bioinformatics, translational medicine who helped ensure that the methods seem able and work well, and the biology team who's really helping move this program forward and create those next set of molecules that we're interested in. And with that, next slide, I just want to thank all of you for listening and happy to take any questions.
Alex. Hi, if you use that model to find the most diverse set of plates for initial hit screening, can you then use that same model to look for the most similar plates to what you've already gotten hits on for optimization?
Yes. So that's the idea, right? We can then utilize to do SAR by catalog, where we have an idea of what is present, what is similar, and try to acquire those compounds, see which directions we can really the features that we're interested in.
Do I see more questions? Allow me some time to walk over.
Thanks for the talk. Just wanted to confirm something. So the reason that you're doing all this is because you can't pick which compounds to put on what plate?
Yes. So the plates are prefixed.
Why are they prefixed?
So they're with our partner, the vendor has already plated the compounds, so we can't actually mix them in a new way together.
I see.
Yeah. So it's kind of just a restriction that we have based on the agreement, on the collaboration agreement that we have with this partner.
Thanks.
More questions? Yes.
So from the drug discovery perspective, I thought the goal is just to find the best molecule that binds the protein, and then that's it, and you move on to the validation. So is it necessary to calculate the similarity or divergence of the plate?
Absolutely. So our risk here is that if you're just sampling very similar molecules and that class of molecules doesn't hit, then you're basically wasting sampling space. You're sampling kind of the same thing over and over again, and we want to avoid that. We want to try to branch out as far as we can to see what are the different spaces that where we can get hits. Can we explore multiple of them and create series out of multiple directions to see what types of properties we get and kind of take the best one going forward off of that.
Oh, I'm sorry, one more.
Thank you for presentation. It was a great presentation. Do you include any clinical data to this discovery, like based on the drugs clinical?
So we include clinical data for the gene target discovery piece, not for the chemistry discovery piece. And in general, for the chemistry discovery, we try to do it in the most unbiased way possible, just looking at what we believe are the molecules at hand, rather than trying to actually fit it into a binding pocket with some a priori kind of biasing towards what types of molecules we want to go for. So